Okay, so I'm going to just sort of tell you a bunch of little stories about our microbiome research at FDA over the past few years. Starting off with just the concept of the Pure Food and Drug Act itself. You know, in 1906, written, eventually leading to the formation of the FDA, at a time when there was absolutely no concept of a human microbiome, or even less so of a food microbiome. So, you know, this is evolving very quickly, and as you can see, it's incredibly complex. The ecology of nutrition, the ecology of pathogenicity, tracking pathogens through these really complex environments, complex. Honestly, it's like sometimes you feel so empowered and then the next day you're completely humbled again. And actually today I'm feeling extremely humbled because we just got back some results that I thought were going to be groundbreaking and fantastic that I'm adding to this, but they're just incredibly confusing. So, but there was a moment at our FDA Science Forum recently when I was really inspired because Eric Lander was talking, and he was talking about just the correlation with all the links to human disease that were starting to come about through all of this next-gen sequencing, getting more and more sophisticated, our bioinformatics getting more and more sophisticated, and how, like, you know, we were puttering along, linking a disease to a gene for years and years, and then at some point it just, you know, went through the roof, and now we're really tracking all of this stuff. And so that was so exciting to me, and I was like, okay, I just got to get my bosses to buy me the NovaSeq, the next Illumina that can do like 10 times as much. It only costs $1 million, and we'll be able to figure out everything. So I was sure that that was going to work, and that we were going to be able to draw lines in agriculture and food safety in the same way we're starting to do, you know, for the human microbiome, which none of this really needs much explanation in this group. So I'm going to move forward. So this is what we're facing. We're looking at this whole food pyramid in a completely different light now. And so our contribution to that, you know, ranges from trying to examine the pathogens in that, trying to just characterize what a baseline is, what's the variability in baselines, is there a core microbiome of any food, what's it influenced by? Much of the focus of our group, many of you are probably very familiar with Genome Tracker. And the concept, I'm just sort of introducing these trackers as I try to lead to what we're going to try to do microbiome-wise with our trackers. But the concept of Genome Tracker is quite simple. You know, you get as much of the genomic information you can about a single genome, and that allows you to make really high-resolution inference, you know, a really precise traceback of its closest known relative. And so this gives us the, you know, the resolution and the power that Genome Tracker has already sort of demonstrated, and it's even been used in court, how well this works. So this is a single genome, you know, largely Salmonella, Listeria, and this is also now being applied to plants, nutraceuticals, plant products. This is being led by Sarah Handy, Dr. Sarah Handy, and the concept is the same. Just take the chloroplast. Many of the barcoding regions or the things that have been used to identify plants have been regions within the chloroplast. So at this point, why not just sequence the entire thing? You know, they're barely over 250,000 bases ever. So this is also a project that's been going on at CFSAN, the Genome TrakrCP. And we did publish a paper where we were able to distinguish between really closely related species of Echinacea. So that was, you know, that's where you break down always, like the closely related species, and it's working very well. In the same way it's working for Genome Tracker to distinguish between serovars, closely related serovars of Salmonella. So, and as I just mentioned, the concept is the same. Just take all the information you possibly can, and you can make these, you know, precise inference about relatedness. So this is part of that crew. That's Sarah Handy. I guess she's on your right. And it's interesting, the camu mention got me really excited because I've studied medicinal plants for years and years with a long-term colleague, Dr. Jim Duke, And we've been using his garden for some of the chloroplast work that Sarah's been leading because he has so many medicinal plant species there. And so we definitely did a lot of work with Camu Camu in the past, and he'll be really excited to hear about this new research on that. So this is the team that's been working on the Genome TrakrCP. That's Jim Duke's Green Pharmacy Garden in Maryland, where we go and sample medicinal plants. So basically that introduction is just sort of to lead us to what we're trying to do with metagenome tracker, which is something that we're just getting started. And the idea, is the same. Take all of the information that we possibly can to create a fingerprint. And that fingerprint is a combination of so many things, archaeal, viral, fungal, bacterial, parasitic, protist, everything that we possibly can, with the idea that maybe we can trace the origin of different food commodities. The idea is that we use this for the farm-to-fork continuum to really identify places where a pathogen can enter, where spoilage can occur, where, what's a baseline, when are things safe and when are they not safe. We started doing this. Our Arabidopsis was, for food safety, microbial ecology, was the tomato, because there had been a lot of tomato salmonella outbreaks on the eastern shore. So we wanted to start with a baseline. This was one of our first papers in 2009, a baseline survey of the anatomical microbial ecology of an important food plant, tomato. So we just kind of characterized it by organ. And then we sort of characterized also just sort of niche-specific, what's living where, and when is that important and when is it not. And characterizing this crop when it's grown in different environments. More of the outbreaks seem to be associated with tomatoes grown in the eastern United States. So we wanted to, profile what the microbial ecology of a tomato grown in the east looks like compared to the west. And it's really different. And this actually sort of led to the identification of a biocontrol that is in the flora of the west and also in the east as well, but in less abundance, and has an antibiotic activity against Salmonella. It actually kills it. I'll tell you a little bit more about that later. Anyway, so one of the really exciting ways that we've been using, metagenomics, microbiome research, is to look at the methods that we have not changed in 50 years, to recover our pathogens, and to try to fine-tune those so that we can identify something as soon as possible, so that food doesn't sit there and spoil, so that people, don't, we get it wrong, it was a tomato, not a pepper, and like all the tomatoes are destroyed. Different things like that. Papayas right now, I think it would be amazing if we could identify very quickly sources of things, fingerprint them. So the way to do that is to really streamline our own methods. So we first started doing this, and this was like the case that just made me think, okay, we're there. We took, there was an outbreak of listeria in ice cream, and we had a look at all the enrichments. What you're looking at here is hour 0 is on the top, and you move down 4 hours. Every 4 hours, we characterize what happens when you do the first 48-hour recovery of listeria. What's going on in there? What is listeria competing with? How well does this work between these different methods? And it was nice to see that our methods do work very similarly, the ISO, the FDA, and the USDA. They're very similar. And then at the end, you see those large bars of all listeria. And so this here, that's just the same image done with 16S. The other one was shotgun. And another concept here is that typically, those first 48 hours are just the beginning. Then you plate it. Then you pick a colony. Then you grow the colony. Then you extract the DNA. Then you have your genome. But with this approach, what we could do is go straight from that shotgun DNA to Cerevar identification. And so that saves us. So you're looking here like maybe just in, what's the actual pointer? There we go. So somewhere in here, for example, we can do shotgun sequencing and get the exact same resolution that we could get with an individual isolate of listeria that would have taken 6 days longer. So we tested this on, this is the tree of the outbreak. All of the outbreak strains of listeria actually this is an abbreviated one, from the NCBI tree now, and all of our different hours where we grew it. So this cuts it in half by day. I mean, this cuts off 6 days practically. So we were so excited that this can then be applied to every commodity. We can do this for every commodity, characterize what the obstacles are, figure out exactly what time we need before we can get the strain level information right out of the metagenome of the enrichment. However, it's a lot more complicated than it was for ice cream. So we've been working on papayas recently. There have been a lot of papaya outbreaks, and this has been super exciting opportunity for us to potentially fingerprint a contamination, because we have 3 different papayas from 3 different outbreaks. One where there were 4 serovars of Salmonella, one where there were 2, one where there was 1, and they're all different. So can we use this to identify, did it come from a truck? Did it come from, the field? Did it come from chicken manure? Can we actually use this to fingerprint it? And then how quickly can we get the genome right out of that metagenome enrichment? Quasi-metagenome would be better, since metagenome refers to culture independent more truthfully. So we started growing these, and we've been doing this. We have, and so this is what this looks like, though. We grow it following the BAM, just doing the typical enrichment that you would do for Salmonella growth. And this is what that looks like. If you're looking, here's after 16 hours. This is the Salmonella down here. Here's after 20 hours. Here's after 24 hours. Then another 24 hours in RV and TT. These are the selective medias, where in Listeria, we were starting to see Listeria from ice cream. Perhaps Listeria from cantaloupes would be just as confusing. But we were starting to see so much Listeria genomic data that we had the serovars right here. So this was frustrating. I mean, maybe if we had the $1 million machine, we could actually get the whole genome of this thing. But it's in such a low abundance, in the background flora, that we can't quite get it yet. We still rely on those additional medias that are so selective for Salmonella, which was really disappointing to me. And so we've tried it a bunch of different ways. This is examining the TT again at 42 degrees, a negative sample and a positive sample, and again, we're seeing these similar things. We had so much Pseudomonas when we grew it at 30, 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 The way that we organize it, like pairing NIH researchers with FDA researchers. So please email me if you're from FDA and want to be part of that. These are things that will be mentioned in our microbiome working groups, and I'm not going to mention the chief scientist proposal right now, but that's all. So if anyone has any questions, thank you.
Thanks, Andrea, although I've heard this talk, I think this is the second time except for your new data. It's really very exciting to see what's going on. I do have a question in terms of this air plant influence. Have you considered insects in that?
Actually, we did. We did an insect exclusion study that I think has just been published. Sarah Allard led that. So she just partitioned off with netting blossoms, and then did the microbial ecology of the blossoms that were allowed to be visited by insects and those that were not. And it was difficult. Again, it was very complex, and there was just greater variability in the ones that could be visited by insects. But there weren't like specific taxa that we could tie to either treatment. But yeah, that's a great question, and Sarah led that and did a really nice job with it. I can send you the link to the paper.